from today, we are diving into the fascinating world of the gastrointestinal system to explore how our bodies break down food, absorb nutrients, and power every single cell. Welcome to the Pathologist Canvas by Dr. Nikki Susan Thomas. So, after a discussion on the cardiovascular system, we're back with another very important topic and very important subject of physiology, that is the gastrointestinal system. So, in this topic, first of all, we're going to see what the digestive system is and what is helping in salivary secretion. All right. So, let's have an overview of the digestive system. So, what is digestion? Digestion is the process by which the food is broken down into simple chemical substances that can be absorbed and used as nutrients by the body. So, this is a term that is familiar to each and every one of us. And most dietary substances cannot be utilized as such. They must be broken into smaller particles so that they can be absorbed into the blood and distributed into various parts of the body. So, the digestive system is responsible for these functions. So, let's learn the functional anatomy of the digestive system. So, in this diagram, you can see, or in this image, you can see various organs like the mouth, the salivary glands, esophagus, liver, gallbladder, etc. So, we're going to learn the functional anatomy. The digestive system consists of the gastrointestinal tract, which is also known as the alimentary canal or the alimentary tract. Okay, and along with this, it has also got the accessory organs. So these accessory organs aid in digestion and absorption. The GI tract is a tubular structure extending from the mouth. So from the mouth it extends to the anus where the food is given out. So approximately the entire length is around 30 feet and it opens to the external environment at both the ends, that is, both at the mouth as well as at the anus. The opening is externally. All right. And now we have to see what are the primary digestive organs and what are the accessory organs or the helping, aiding organs. So let's see what are the primary digestive organs. The primary digestive organs are the mouth, the pharynx, the esophagus, the stomach, the small intestine, and the large intestine. Whereas the accessory organs that helps these primary organs in digestion, they are teeth, tongue, salivary glands, exocrine part of pancreas, liver, and the gallbladder. Even though we call them accessory organs, they are not less important. They are equally as important as the primary organs that helps in digestion. All right. Now let's see what the wall of the gastrointestinal tract is made up of. So there is an innermost layer that is known as the mucus layer. Okay, and this layer is the innermost layer and it comprises of the epithelial lining in contact with the GI contents. Then the lamina propria of the connective tissue and the muscularis mucosa of the smooth muscle fibers. This is what the mucus layer or the innermost layer is made up of. Now, the next layer is the submucous layer. The submucous layer is present in all parts except all parts in the sense it is present in all parts of the GI tract except in mouth and pharynx. So it contains the loose collagen, the elastic, and the reticular fibers. Then also the blood vessels and the lymphatic system or the lymphatic vessels, as well as the nerve plexus. So this is a very important layer. And the next layer is the after the submucous layer, we have got the muscular layer. So the muscular layer is the layer that contains the muscles. So the lips, cheeks, and pharynx contain skeletal muscles. So that's why we can voluntarily smile, pout, etc. So esophagus has both skeletal and smooth muscle. That is why it has got involuntary action as well. And stomach has three smooth muscle layers. So the inner layer, so that is a bit different here because we have to consider the layers or the smooth muscle layers of stomach as well as the smooth muscle layer of the intestine. Okay, so don't get confused here because the smooth muscle layer of the stomach is different from the smooth muscle layer of the intestine. So the stomach has got three layers. I'll write S for stomach and I for intestine. So the stomach has got three smooth muscle layers. That is an inner oblique layer, then a middle circular layer, and an outer longitudinal layer. And the intestine has got only two layers, and they are the inner circular layer as well as the outer longitudinal layer. So these are the muscular layers present in stomach as well as the intestine. Now we're moving into the last layer of the GI tract, that is the fourth layer, that is the serous or the fibrous layer. So the outermost layer and the serous layer or serosa covers the stomach, the small intestine, and the large intestine, and the fibrous layer or fibrosa covers the pharynx and esophagus. So we cannot say that the GI tract outermost layer is serous layer, or we cannot say that the outermost layer is fibrous layer. It depends upon which part of the GI tract that we are referring to. So as I said just now, the serous layer is the outermost layer when it comes to the stomach, the small intestine, as well as the large intestine. Whereas when we talk about the pharynx and the esophagus, which is the outermost layer, the fibrous layer. Okay, so that is the difference. Clear? Now we're moving into another important topic, that is the nerve supply to the gastrointestinal tract. Okay, nerve supply. So there is both intrinsic nerve supply as well as extrinsic nerve supply. Intrinsic as well as extrinsic nerve supply. So the intrinsic nerve supply is also known as the enteric nervous system, or it is intrinsic nerve supply is managed by the enteric nervous system, whereas extrinsic nerve supply is a different entity. So the enteric nervous system runs within the GI wall from the esophagus to anus, forming two major networks. So it forms two important networks. And the first network is the Orbax plexus. So this is actually I will write it separately. There are two networks that the enteric nervous system makes, and the first one is the Auerbach's. It's Auerbach's Auerbach's plexus, which is also known as the myenteric plexus. And we have got the second network, that is the Meissner's plexus. Okay, so this is also known as the submucous plexus. So, what are they? The Auerbach's plexus or the myenteric plexus is located between the inner circular and the outer longitudinal muscle layers. So, this what the Auerbach's plexus does is it regulates the GI movements. So, it can either have excitatory action or excitatory fibers can have a separate action, as well as inhibitory fibers can show a different action. So the excitatory fibers secrete some excitatory neurotransmitters like acetylcholine. Just remember these acetylcholine and all the other substances as just chemical substances. So since I use the term excitatory, that means which would help in excitation. That means here the GI movement will be more. It's excited, so it will show more movement. Whereas inhibitory means it is going to inhibit the movement. That means the movements of the GIT will be inhibited. So that is the difference. Fine. So the excitatory neurotransmitters here or the chemicals, just remember them as chemicals because for a first year physiology student, you might face it difficult when you hear such terms like neurotransmitters, etc., because we have not covered the topic yet. All right. So acetylcholine, then the next chemical is serotonin. Then we have substance P. Then we have certain inhibitory fibers which secretes VIP, that is vasoactive intestinal polypeptide. So it is inhibitory. Then neurotensin, and another substance known as the enkephalin. So since these are inhibitory, they will inhibit the regulation or inhibit the movements of the GI tract. Next, network is formed by. So what am I talking here about? I am discussing here about the intrinsic. And first of all, the major topic that we are discussing here is the nervous supply of the GI tract. So, out of these nervous, or out of these, what comes under this total nervous supply is the intrinsic as well as the extrinsic nerve supply. So the intrinsic nerve supply has got two networks. That is what is these Auerbach's plexus as well as these Meissner's plexus. Fine. So don't get confused here because here we use so many new terms, so it might be confusing to you. So as I said in the previous lecture of cardiovascular system, what you have to do is you have to see this class and go back and read your textbook. So you'll get a more detailed idea of what I am teaching here. All right. So I hope you have understood Auerbach's plexus. Now it's time to move on to the next thing. Yeah, that is Meissner's plexus. So Meissner's plexus is located between the muscular and the submucosal layers, and it regulates the secretory functions. Okay. So they are not concerned with the movement of the GIT as the Auerbach's plexus is concerned about. Here they are more concerned about the secretory action. So it regulates the secretory functions and causes constriction of the GI blood vessels. Constriction. Okay. So that's about the intrinsic nervous system. Now we have the most important extrinsic nervous supply. That is another topic. So the extrinsic nerve supply can be either through the sympathetic fibers and the parasympathetic fibers. I think it would be good if I can discuss with you all the nervous system next after covering the GIT, because since you might not be much aware of what the sympathetic fibers or parasympathetic fibers are. So for now, just understand it as types of fibers. So we have intrinsic as well as extrinsic nervous system that is helping the GI system manage its secretions as well as its movement. So under the intrinsic, we studied there are two networks, that is Orbax plexus as well as Meissner's plexus. And the intrinsic system under Orbax had excitatory as well as inhibitory neurotransmitters, and it helped in the regulation. So when there was excitation, the movement increased, and when there was inhibition, the movement reduced. And the second network was the Meissner's plexus. So in this diagram, can you all appreciate here this particular, just look at the diagram. So that blue dots that you see is the Orbax plexus, and the Meissner's plexus are the green dots that you see within the Orbax plexus. Okay, so this is how the structure of intestinal wall looks like. And then the second network, that is Meissner's plexus, was more concerned with the secretory function and not the regulatory function. And then the second type of nervous supply is through the extrinsic nerve supply, and it has got both sympathetic fibers as well as parasympathetic fibers. So the sympathetic fibers, each of these sympathetic as well as parasympathetic fibers will have preganglionic as well as postganglionic fibers. So preganglionic means before reaching a particular ganglion, the fiber is known as the preganglionic fiber, and the fibers after going through the ganglion, that is known as the postganglionic fibers. So the preganglionic fibers arise from the lateral horns of T5, that is thoracic fifth vertebrae, to the L2 spinal cord, that is lumbar 2, and they pass through the sympathetic chain without synapsing, and they terminate in the celiac and the mesenteric ganglia. Since this might go beyond the head of first year BDS students, just understand that the preganglionic fibers arise from the lateral horn of T5 to L2, and then they terminate in the celiac and mesenteric ganglia. I hope my MDS friends have already understood what the whole idea is, because the preganglionic fibers arise from the lateral horns of T5 and L2, and they pass through the sympathetic chain without any synapse, and then they terminate in these two ganglia, that is the celiac and the mesenteric ganglia. I hope you have understood this. And the postganglionic fibers will innervate the entire GI tract. Post-ganglia. That means the fibers after moving out from the celiac and mesenteric ganglia, the postganglionic fibers will innervate means they supply the entire GI tract. And what will be the effect here? The effect is that it will inhibit the GI movement, and it also reduces secretions, because here the chemical, I already told you, there are several neurotransmitters. So here the secretion is reduced because the neurotransmitter that is released here is noradrenaline. Okay, noradrenaline, and then it also has another function, that is it constricts the sphincters. So, what are the effects of the sympathetic fibers? The preganglionic fibers, they will end at the celiac and mesenteric ganglia. And then what happens? The postganglionic fibers will show these effects. That is, they will inhibit the GI movement, they will reduce the secretions, and that is because of this noradrenaline. And also, one more effect is that it will constrict the sphincters. So, which means altogether the sympathetic system has got an inhibitory action on the GI tract. Now, talking about the parasympathetic system. So, the parasympathetic system is a system which supplies the supply to the mouth and salivary glands. So, via the facial and glossopharyngeal nerve. For first year students might have already studied about the cranial nerves in anatomy. So we know that the facial nerve is the seventh cranial nerve and the glossopharyngeal nerve is the ninth cranial nerve. So the parasympathetic fibers supply via the facial as well as the glossopharyngeal nerve, and they will supply the esophagus, the stomach, the small intestine, then the upper large intestine, everything via not the facial and glossopharyngeal nerve, but via the vagus nerve. So the supply of the parasympathetic system depends upon the location once again. So the facial and the glossopharyngeal nerve supplies the mouth and the salivary glands. That means the parasympathetic fibers supply to the mouth and the salivary glands via the facial and the glossopharyngeal nerve. Whereas when you consider the other areas lower to the salivary glands, which are those areas, we have the stomach, we have the small intestine, we have the large intestine. So all these are supplied by the vagus nerve. So vagus nerve is the 10th cranial nerve. Fine. And we have the lower large intestine that is further down the upper large intestine. So this lower large intestine, I will write it as LLI, lower large intestine, is supplied by the pelvic nerves. That is via the sacral nerves S2, S4. So the pelvic nerve supplies it. So all the preganglionic fibers will synapse in the myenteric and submucous plexus. And the effect is what is important here. Effect. The effect is that it will accelerate the GI movements and also it increases the secretions. Why is it? It is because the chemical that is released here or the neurotransmitter released here is acetylcholine. So please keep this in mind. The sympathetic system produces noradrenaline, so it is inhibitory, whereas the parasympathetic system produces the acetylcholine, which is an accelerator, or it is more like excitation that is happening here. Clear? So we have covered a very important topic, the nervous control of salivary secretion. That is a very important area. Okay, or altogether the it's not nervous supply of the salivary secretion as such, because for salivary secretion we have got much more to learn, but this was the overall nervous supply of the GI tract. Okay, so this can come as an essay or a short note, or it can come as a short essay as well. Now we are moving into saliva in detail. So that is another session. Maybe you can take a small break and then resume back to studies. Now, after this small break, we're back to learn more about salivary secretion. So, let's learn the functional anatomy of the mouth. The mouth is formed by cheeks, lips, and palate. So, it encloses the teeth, tongue, salivary glands, opening anteriorly through the lips and posteriorly through the fauces. Right? Through the fauces, posteriorly it opens out into the pharynx. So the digestive juice that is present in the mouth is saliva that is secreted by the salivary glands. Now, what are the functions of the salivary glands? The functions are definitely to ingest the food materials, for it helps in chewing, then it helps in appreciating the taste, right? And it has also got a role in speech, and also socially it helps us a lot, because dry mouth without saliva causes a lot of problems like caries, etc. So what are the various salivary glands? Saliva is secreted by three pairs of major salivary glands, and also there are several minor salivary glands. So the major salivary glands are the parotid gland, the submandibular gland, as well as the sublingual gland. The parotid gland is the largest of the salivary glands. It is located at the side of the face, below and in front of the ear. So just visualize this as I tell you. It is present below and in front of the ear, and it drains via the Stenson's duct. The duct of parotid gland is the Stenson's duct. So where does the Stenson's duct open into? It opens inside the cheek against the upper second molar tooth. So this is the region where the Stenson's duct usually drains. Now moving into the next one, that is submandibular gland. It is located in the submandibular triangle, which is medial to the mandible, and it drains via the Wharton's duct. It opens at the side of the frenulum of the tongue, or the frenulum of the tongue, at a structure that is known as the caracula sublingualis. Then we have sublingual glands, which are the smallest of major salivary glands. In the mucosa of the floor of the mouth, it drains via the ducts of Rivinus, which are a group of 5 to 15 ducts. And the largest of these ducts of Rivinus is the Bartholin's duct. It opens near the submandibular duct. So the ducts of each of the salivary gland is important. For parotid, it has got the Stenson's duct. For submandibular, it has got the Wharton's duct. And for sublingual, it has got the ducts of Rivinus, and the largest of it is the Bartholin's duct. We have also got various minor salivary glands. Okay, various minor salivary glands are present, like the lingual mucus glands, then the lingual serous glands, the buccal glands, as well as the labial glands. And also one more thing, sorry I forgot, that is the palatal gland. So the lingual mucus gland is present at the posterior 1/3 of the tongue, which is behind the circumvallate papilla. So on the tongue, we have got the circumvallate papilla at the posterior 1/3. So this particular lingual mucus glands are present behind the circumvallate papilla and at the tip or margins. And the lingual serous glands, they are near the circumvallate and the filiform papillae. The buccal glands are between the mucous membrane and the buccinator muscle. That is how you can remember the name. And the labial glands are beneath the mucous membrane around the orifice of the mouth. That is around the opening of the mouth. And the palatal gland is beneath the mucous membrane of the soft palate. So usually the salivary glands can be classified into salivary glands. I'm writing it as SG. So it can be either serous, it can be mucus, or mixed. So serous glands are predominantly, they usually secrete thin, watery saliva. So the example of serous is parotid, as well as lingual serous. So parotid is a major salivary gland and lingual serous is a minor salivary gland. So they secrete watery saliva. That is the peculiarity of serous glands. And coming to mucus glands predominantly secrete thick, viscous, mucin-rich. From the name itself, you understand they are mucin-rich saliva. So the example of this is the lingual mucus, buccal, and palatal. So from that, it is well understood that most of the minor salivary glands come under this section, not the major. And when you talk about mixed, we have both serous as well as mucus cells. Example is the submandibular, the sublingual, as well as the labial glands. So we have got my major salivary gland, that is submandibular, then minor like sublingual is also one of the major salivary glands. Labial is a minor salivary gland. So both major as well as minor salivary glands are present here. Now let's have a discussion about the properties and composition of saliva. So this is the composition of saliva. The saliva has got 99.5 percentage of water and 0.5 percentage of solids. Now, out of the 0.5 percentage of solids, we have got organic substances as well as inorganic substances and some amount of gases. So the organic substances are enzymes as well as other organic substances. So this is, if you can learn this particular method of writing the composition, you can write the same thing for gastric juice, same thing for pancreas. I mean, it. I do not mean that you can write the same list of items given here, but the way it is shown, like water, how much percentage, solids, how much percentage. Then again, from solids, you can divide it into organic substance, inorganic substance, and gases, and then organic substances into enzymes and other organic substances. So, like that, the headings can be the same. It can be applied to gastric secretions as well as pancreatic secretions, which we will discuss in the next class. So, coming back to this classification, and always remember it is best to draw flowcharts and such illustrations for your exam, because it makes it easy for you as well as for the uh, invigilator who is correcting the paper. Fine. So the organic substances include enzymes like amylase, maltase, lingual lipase, lysozyme, phosphatase, carbonic anhydrase, and kallikrein. The other organic substances are mucin, albumin, proline-rich proteins, lactoferrin, IgA, blood group antigens, free amino acids, non-protein nitrogenous substances, urea, uric acid, creatinine, xanthin, hypoxanthin. And inorganic substances includes a lot of elements like sodium, calcium, potassium, etc. And gases are oxygen, carbon dioxide, and nitrogen. And normally it's given there. See, glucose is absent in saliva, but it is found in saliva during diabetes mellitus. So the presence of sugar or glucose in urine is an abnormality. That is because of diabetes mellitus. Fine. So this is the composition of saliva. Now, talking about the properties of saliva, the volume of saliva is usually approximately it is 1 milliliter per minute. That means it is around 1000 to 1500 ml per day. And out of this, parotid produces 25 percentage of the saliva, submandibular produces 70 percentage, and sublingual produces 5 percentage. So submaxillary produces the major share of saliva. Next property is its nature. It is slightly acidic with a pH of 6.35 to 6.85. The specific gravity is 1.002 to 1.012. It has got a hypotonic nature, and its composition, again, you have to mention the water constitutes around 99.5 percentage, whereas solids constitute only around 0.5 percentage. So, what are the basic functions? You can write the functions of saliva by just looking at the composition of saliva, because most of the things here, for example, the digestive function, that is one of the most important functions of saliva, digestion. Just look at the enzymes here. This will give you a proper idea of how saliva helps in digestion. Salivary amylase, that is the first enzyme given there, this will convert cooked starch to starch that is present in the food is converted to dextrin and maltose. So this should happen only at an optimum pH of 6. Then we have maltase. Maltase converts maltose to glucose. Then let's take another example, that is lingual lipase. So lingual lipase hydrolyzes the milk fat triglycerides into, so triglycerides are converted into fatty acids and diacyl glycerol, which are the broken down products of the triglycerides. So, like that, the first three are helping in digestion. Then another one is, see, it's given there, lysozyme, right? So, what is the function of lysozyme? Saliva has got various properties like cleansing and protective function. So, it rinses the mouth and teeth of any food debris, and this particular lysozyme helps in killing, or it has got an antimicrobial property, and it can kill various microorganisms like staphylococcus, streptococcus, and brucella. Fine, that is the function of lysozyme. And saliva has also got a role in speech because it can moisten and lubricate. So, digestion is one function. Then we have the cleansing and protective function that I told you just now. Cleansing, protective, antibacterial action. That has got an action in role of speech, and also excretory function because it saliva excretes mercury, potassium iodide, lead, thiocyanate, and even some viruses which are highly dangerous to humans. So that is also helped by saliva. And the regulation of body temperature, usually this is most important in case of dogs and cattle because they show drooling of saliva. Okay, you have seen, right? Dogs panting and saliva drooling out from one corner of their mouth. But in humans, the sweat glands play a major role in regulating the body temperature. And also, it regulates the water balance in the body in the form of sweat, inducing thirst, and prompting water intake, etc. And other than this, it helps in preparation of food for swallowing or lubrication, facilitating chewing, then rolling of the bolus, everything. So definitely it helps in swallowing. Then appreciation of taste because of the taste buds, and also it will stimulate the taste buds, enabling taste perception. So these are the functions. And the digestive enzymes are very important, especially the salivary amylase, the salivary maltase, and the lingual lipase, which I have already discussed now. And these three enzymes will need an acidic activator that will help in splitting the major component into simpler substances. Okay. And there is one more important topic to be covered here, which I will cover in the next session because that is the regulation of salivary secretion. So, in today's session, we learned about how the entire GI tract looks like, what is its composition, what are the layers, and what is the innervation or what is the nervous supply to the GIT. And we also saw about what saliva is, what is the composition, what are the properties, and functions of saliva. And the method by which, or the regulation of salivary secretion, will be dealt in detail in the next class. And the next part two, we will also discuss about another important thing, that is about how gastric secretion occurs and also how pancreatic secretion occurs. So that will be a class exclusively wherein we will learn about the secretion, various important secretions, and you will have many more important terms coming up. So please see this entire session and then go back and read your textbook and make you familiarize with the topic so that you will understand the innervations better. Because here we had terms like Orbach's plexus, Meissner's plexus, etc. So for that, you should have a thorough idea about this classification, how the nervous system is divided into intrinsic and extrinsic. And out of intrinsic, I told you there are two pathways, Orbach's and Meissner's. And in extrinsic, we had again two pathways, that was the sympathetic fibers and the parasympathetic fibers. So, what are their chemical mediators? Like for sympathetic, we have noradrenaline, and for parasympathetic, we had the acetylcholine. So, all that you need to make thorough. Okay. And so happy to see you all again. And if you have any doubts, as I usually tell, please post in the comments. And I'm very happy that some of you are messaging me, asking doubts, and please continue doing the same. And you can reach me at Dr. Nikki Susan Thomas. at my Instagram page where I'll get back to your doubts. Okay. So that's about it. Thank you. Have a great day.